Hey guys, these are the review questions that we had for kind of what happened on the exam. The ones that you guys see in front of you. So the energy requiring steps of glycolysis are 1 and 3, and the rate limiting steps are 1, 3, and 10. Yup, but we'll get into that a little bit in a second. What do they mean by residues? Residues is referring to which of the amino acids in that given sequence show up where they do. Okay. Sometimes they use the term motif whenever we're talking about biochemistry, specifically what the typical chemical properties are of the area we're looking at. But residue is simply referring to that's what it actually is. It's a glycine, it's an alanine, it's a serine, glutamine, etc. What is a PPI? That's a pyrophosphate. So that's literally two phosphates bonded together. So whenever they both get ratcheted off, like whenever we're going through and performing glycogen synthesis, that's going to free up more energy. And then we're obviously just the PI is simply referring to as inorganic phosphate. So what does the symbol that looks like an A before 1,6 linkage mean? And this is in reference to the bonding pattern for glycogen. So 1,6 being a branch, alpha 1,4 being your typical straight in a row. The alpha is actually referring to the configuration of those bonds. So the two that we see typically in nature are alpha and beta. So it's essentially, is it a down or is it a cross and going up? Well, alpha is the one that we're going to see in the human body. Plants, that's why we see beta, and the body enzymatically can't break it down. That's why it turns out cellulose is fiber, and, well, fiber has those certain effects upon your digestive tract, producing something, if you will. And then I know that ATP synthase, blah, 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 blah. Now, yes, understand that that is going to be the final piece of the electron transport chain. That's where we're going to be producing energy because of all those protons that we've built up in that inner membrane space. That's going to create that massive gradient that then pushing those hydrogens back through is going to in turn synthesize ATP from ADP and 1-phosphate. But keep in mind that all the steps that come up before it are important for this to occur. If those steps don't occur, then it turns out we're not going to be producing this. And then Randle cycle at the end of the day is basically just a facilitator full of competing substrates. I'm going to guess what you're getting at is, don't think of it as a facilitator, but it's a way of explaining how the body avoids running both processes at the same time. So that's where we're going to be utilizing both fuels at the same time, but we only have so many enzymes to break down those fuels. So it's not exactly a futile cycle like would occur if both glycogen synthase and glycogen phosphorylase were activated, aka trying to build glycogen while we're also breaking it back down. However, it is going to be inefficient. So just to first talk about the initial question with glycolysis, here's just that nice little flow chart. So you can see step 1 with hexokinase is going to require energy, as is step 3 with PFK is going to require energy. So both of those are going to be rate limiting steps. And then finally, pyruvate kinase, the final step before pyruvate's created, which actually creates ATP, is going to be that other rate limiting step. And that's actually important because then when we come to the Randle cycle, we can see how the simple ability to essentially shunt off pyruvate's conversion one step later into acetyl-CoA is how we're going to have that biofeedback mechanism inside of the cells of our body, so that we're either relying upon fatty acids via beta oxidation for energy, or we're relying upon glucose via glycolysis as the means of producing energy. Now, this is simply by the products via allosteric modulation are going to be inhibiting further creation of those different substrates that are going to enter into the citric acid cycle from glycolysis. And then I also want to point out the other cycle to make sure you remember is the Cori cycle. And this is specifically how the body is going to handle excessive amounts of typically lactate that's going to be produced during exercise, which is then going to undergo gluconeogenesis inside of the liver. And at that point, it will then be able to be converted back into glucose and then possibly released again in the bloodstream if it's needed, or then stored in the form of glycogen until it's needed at a later date. Hope this answers all of your questions, guys. Good luck on the exam tomorrow. You're going to do great. Thanks.